My name's Andrew. First of all, a reminder to say thank you to Andy and Carly for giving me a good reason to be back in Bath. And for the last 20 plus years, I've always worked on therapeutic antibodies, so perhaps here I'm a little bit of an imposter, but I do think there's a lot to be learned from the therapeutic antibody people engaging more with the tool reagents, because it's all the same fundamental science. If I was really down with the kids, this should be a personal perspective, and it should be on agentic AI, but we're not going there today. But it's a lot of fun to be here. So just as a kind of big picture overview of antibody tech for drug discovery. So the figure on the right is basically the paradigm most of us think about. Biologists have key insights to some aspect of biology and think there's a therapeutic hypothesis to follow, and off we go making antibodies. That antibodies obviously moved mouse, chimeric, human. There are two brilliant Nobel Prizes in the 80s to Jern Kohler-Milstein, and in 2018 to Arnold Winter, and I've forgotten the third name, using human antibodies and other protein engineering techniques. But all the time, we're making molecules, we're advancing them preclinically, clinically, in terms of manufacture, and there's always this backwards and forwards. Are we actually learning anything? And that's kind of driven this figure on the right. In the 80s, all this tech got us into what we call the biotech revolution. But now, in the 2020s, we are in the age of multispecifics. There's some brilliant big review papers on that topic. But that's also, and by multispecifics, we're basically saying more than one mechanism in one molecule. So at its most basic, we're in bispecifics. There are some brilliant assets in the clinic. There are 11 marketed products. You can tag things on antibodies. Antibodies can be antagonists, agonists. Antibodies can degrade. Antibodies can do target-independent functions. And then we add on top the age of genAI. Press go and you will get something. It's not actually true. So this is really reflecting on the AI component in all of this. If there's nothing else to take home, if you're going to enter the age of AI and ML, well, we are all entering, you don't get a choice. The big thing to be aware of is, what question are you asking of it? What data have you actually got? And is it high quality data that's relevant to the question and the context you're about to operate in? That's basically what you need to know. We could all go to the pub. No, for drinks. But this is the schematic that many of us will be aware of. Discovery, find molecules, validate it, think about the translation, and head to the clinic early and late. The big breakthrough for AI and ML tech is actually in the clinic. This is a lovely little schematic from Sanofi. They have a really nice kind of, they're really strong in this space, as are many, my former employer and many other big pharmas have great initiatives in this space, but they describe it really clearly. To launch a clinical trial could take you 18 months. Using these various kinds of tech, the ability of, with your large language model, to help recruit patients, to help find the right clinical sites, to analyze the data quickly, can turn around what could be a 1.5 years process, and it might bring it down to 6 months. This is saving a massive amount of time and budget and helping farmers move forward in the clinic to actually get to the very far right, where you're actually achieving patient care because you've got a meaningful medicine to patients. But today, we're right back on the other side in discovery. And in discovery, there are really two categories of antibodies. I work primarily in candidate drugs, at the same time then with Carly in the past, working on reagent tool antibodies. Both are equally valid and both are equally valuable to making medicines, because it's almost to the point today that if you bring your molecule, be it an antibody, forward for its BLA, the regulator will say to you, where's your biomarker? Often those are antibodies. If you haven't got one, they may well say, well, you're not getting a marketing license. They want biomarkers, they want patient selection, and the payers want patient selection. Somebody asked earlier about the kind of differences in scale. For a candidate drug, there are very high hurdles to meet, but they're all just an extension of what's happening in the tool antibody world. And we'll kind of go into some of those in more detail. Each of those little lines, like specificity, in vitro potency, they're all very, very similar to the reagent antibodies. But then there are other categories that Carly alluded to earlier around developability. There's the whole other field of immunogenicity to be addressed. Each of those little lines turns out to be a massive team of people and other specialists that you need. But these antibodies that we tool antibodies for research, for diagnostics, for PK, for ADA, all these bioanalytical assays, they also need suitable QC. And if you're going to take it all the way to the clinic, that needs, again, it's a high bar. And that's why quality is imperative. It's not just a scientific driver. It has implications for all these things around patient safety, success, and reputation. Carly gave a really nice example of her phosphatase or antiphosphate antibody, where the recommendation is don't progress with this particular target because you can't believe the data. And we had lots of other brilliant examples, one from Stefan. Your reputation as a scientist in a company will hinge on how believable your data is. So having orthogonal data sets, which is why actually having to fill in all these boxes for a candidate drug, it makes it more robust. Ulf talked about antibodies being like badly behaved children. In the candidate drug world, I think of them as well-behaved children. Because you've characterized, almost trained them, they will do what you want them to do. It's very, very rare that they don't. There are a few examples, and published examples, where they haven't been well-behaved, but it's really important. And companies' reputations ride on them, which is why from the reagents to the drugs, companies take this very seriously internally. So it's a really, really worthy thing to have these kinds of meetings and conversations. But rather than showing you a particular paper on AI, because there are a pantheon of AI ML models, these are just some of the most topical ones. So diffusion is the technique that Baker wins the Nobel Prize for, well, alongside, I didn't put AlphaFold here actually. So to make it clear, Baker runs a technique that's called based on diffusion. Habis and Jumper, who were the other winners of the Nobel Prize last year, run it, are basically AlphaFold. But even the AlphaFold team, they have AlphaFold 3, AlphaFold Multimer, and now AlphaProteome, which is not available to the public yet. They published that they can't deal with antibodies. But these other methods attempt to. But this was just my view at the end of last month on all these different attributes that antibodies need to achieve to become candidate drugs in a kind of traffic light. So the most successful area is around affinity and potency, because the potency is ultimately linked to affinity. In lead-op, which is a bit like Deborah's talk, she alluded to it, you can do ML, augmented, assisted, co-piloted antibody optimization. That tech works today. It worked from the 2018s, mid-2010s. But it always works in a unique, your antibody, that antigen, not in a general way. So you build a model, you train it, it works on your system, but you need to retrain it to work on another system. The biggest area of success, I think, is developability. So this is how well can you make this antibody? How fit is it to the CMC platform to scale it up? Here, masses of data, masses of progress in research. It's slightly different then when it gets to scale up. The actual, now you're going to get this thing up to 100, 150, 200 mgs a mil. This is challenging, and that's why it's still in amber. There was a really great paper a couple of months ago from Amgen describing their machine learning model in the CMC attributes of antibodies. And just because we are all concerned about the reproducibility crisis, this is a CMC team making, I think they made about 80 molecules at 100, 150 mgs a mil. It's an expensive experiment. They run their study. They get their really high-end mass spec data to characterize them, publish a lot, and they actually say in their paper, Most of these antibodies have been studied by another company, very reputable, Biorad, same endpoint, and they say in their paper, we are acutely aware of the disconnect between the data that we're publishing and the data that Biorad published. They're both really good labs doing really top-end experiments, and it's still not reproducible. It's a confounding problem that still we wrestle with, and still these high-end, big pharma CMC teams are wrestling with how to address it. But to move on from the challenge of the data, it's really good, and it was good this morning that Andrew Bradbury introduced an overview of the progress in the AI antibody challenge. These models, these competitions are good for two reasons. One, they provide new data to the community. And two, you can begin to assess the accuracy, like would you actually have the confidence to pick up and begin to use or spend your valuable R&D budget or research budget on that kind of tech. So there's AI antibody, Adaptive Bio they've had 2 iterations of their competition. They've already published it. Well, it's on bioRxiv. This new one, Bits to Binders, I only saw last week. It's published a result. They're making binders for CAR-T. And then Ginkgo just published this one yesterday or earlier in the week to say we're starting this one on developability. So these are all really good initiatives to try and build improvements and benchmarking in this community. There's a brilliant academic called Charlotte Dean in Oxford. She always has really good one-liners. Her one-liner for this question of benchmarking is, if these techniques can't do a better job than a well-informed scientist with some rules of thumb, you may as well go out and just burn trees, because these things are energy intensive, which is a whole other thing to talk about, perhaps over dinner. But actually, the gap is not the tech. The tech is brilliant. It's all built off the same data. So the gap is the data gap. That's the biggest missing thing. Data is sparse for antibodies. The PDB is brilliant. The PDB is full of protein-protein interactions. I'll not do a quiz. There are about 3,500 solved antibody-antigen structures, most of them to COVID or hemagglutinin. It's very, very sparse when it comes to antibodies. And this is a brilliant paper, again, from Alyssa and Konstantin, PhD students, trying to quantify mathematically how much more data would you need if you were going to ever have a machine learning model that was generalizable for antibodies. And this is that I recommend reading the paper. You need a lot of data, and it ain't out there. Which is why you will see more and more companies prepared to enter federated learning. So a federated learning, all it really means is you hold your data, you're one of these little cell phones down here, company 1, 2, 3, 4. You hold your data behind a firewall, but you all agree to train a model. So you pull that model down from your third party, you train it, you send it back. Everybody does that all the time until you all agree the model's got to this point, then you all bring it back down and test it. Has it got any better? So there are 2 pay-to-play federated learning things. There is Aphorisys, ASIB, which these guys have all bought into, and there's Fate. My claim to fame is I designed the logo, then I had to leave, unfortunately, because I left AZ. But I really hope these do really well. But they're going to have the challenge. How useful is our data? Can we actually get to the same standard? And then just yesterday, Lilly, bless their cotton socks, just launched this thing, TuneLab. TuneLab is a way to access their brilliant antibody and small molecule machine learning models, in the antibody world is to do with developability again, but you can get access to that for free if you're prepared to enter their federated model. And some biotechs have. I think it's a brilliant way to get access to tech that most of us can't afford to operate, but you can in Lilly's model. So let's see how it goes. And finally, people are committing to making new machine learning grade data. So in the UK, the biggest one is OpenBind. They're clearly saving on their marketing budget because that's all their website has. It's just a little ticker. No graphics, no nothing. But then UCL and AstraZeneca have got this successful new prosperity partnership to build AI models and data that's relevant to antibody manufacture. So I hope they do really well. I suspect OpenBind is going to really focus on small molecule protein interactions rather than antibody protein interactions, just because of the scale they hope to do, but we'll see. And finally, this is a discussion again for dinner perhaps. Is there a more credible thing to do with AI? I'm going to propose the mini binder FC. So this is building on what Jonathan said earlier. So up till now, de novo mini binder design. And by mini binder, we mean these little 3-helix or 3-helix beta sheet, hard as brick dust molecules. The design of those was a specialist topic, and we saw Baker's papers in 2022 off diffusion. But now the Carrera lab, which was, again, Jonathan just showed in the last session, some examples of that being tested. It's almost routine. So this is the Carrera Labs paper, which only came out last week, where they claim one shot. But they're claiming success rates of 10 to 100% on this one design, one binder. And this is just some data where they're showing they're getting nanomolar levels. These were the two best ones I chopped out of their bigger figure. But I do think that in our world where we want to make binders to novel proteins out of the proteome, the dark proteome even, where there are no existing antibodies, why would you not use this tech? It's free to access the tech these days from the Carrera lab, and I presume from some of Baker's alpha diffusion stuff. The pros and cons of them, up and down. But as a kind of take-home perspective, AI/ML today doesn't match the well-established antibody platforms we know and love and then spend our time characterizing the output of. However, it is a very useful co-pilot if you have particular questions around engineering existing molecules, be they antibodies, enzymes, domain antibodies, proteins, peptides, it doesn't really matter. You can't train them to do stuff. The gap with all of it is high quality data and reproducibility between labs. That doesn't go away. It's not magic. But I do recommend us as a community engaging more with mini binders. And talking to Jonathan, actually, don't just think of them as mini binders. Stick an FC on them. Then they're bivalent. You get over some of these affinity problems. They could really help in the basic science and diagnostic tool space, I propose. That's it. Time for the book.